All right, everybody. So, welcome back. Today we're going to do chapter 11. We will almost certainly get through all of chapter 11 today, and we'll do chapter 12 on Thursday. So, this is kind of maybe a clue that we're going to start covering material a lot more quickly for the rest of the semester. We have, I think, about 8 more chapters to go through, which sounds like a lot, but like I said to a few people before, we're not covering so much in detail and in depth from each chapter from now on. So, although it's a lot of chapters, it's not an overwhelming amount of material for you. So we're just going to be moving fairly quickly through chapters, though. So please keep it in on schedule with all of the material that's posted on Brightspace and with the course schedule that's posted, so that you're prepared and ready for everything. That's right. Yeah. So we're doing chapter 11 today, and the quiz for chapter 11, which is quiz 9, is due a week from today, next Monday. That's your next quiz. There's also the project update that's due tonight for those of you who haven't submitted it yet. I think those are your big thing. Yeah, that's your one big thing due this week is the project update. And then next week we'll start having quizzes about once a week. I think maybe one week we have 2 quizzes in a week. But yeah, we'll see how it goes. Yeah. So I think that was all I had to say for you so far. I apologize, I don't have your exam grades yet. I'm going to try to have them for you by Thursday. So I will let you know on Thursday how that's going. Does anybody have any general questions about assignments that are due or anything about the class in general before we get started? Yes, no? Okay. Yeah. Yes. So we have quizzes. There's also if there's an optional rough draft submission for the project, which is the Monday after Thanksgiving. If you want to submit a rough draft, you can. It is not required, but if you want more feedback on how your project's going, you're welcome to submit that rough draft also. Yeah. And I just also updated that assignment so that you can submit by your groups if you're working in groups. So if anyone has trouble submitting as a group, let me know. This is the first time I've tried that. So, yeah, not for the one that's due today. I didn't get to it in time, but the other project submissions can be by group. So yeah, if there are no other questions, let's get started with chapter 11. So it is all about enzyme catalysis. And so we're going to do the first section, which is defining and categorizing enzymes. And of course, if I have my pen on, I can't click on this. Okay, so let's just get started by thinking about enzyme catalyzed reactions and kind of definitions. So in this first question, it says delta G for a reaction that is catalyzed by enzyme A is negative and small. It's about negative 0.45 kilojoules per mole. So what conclusions can be drawn about this reaction? So I'd like everybody to take a couple of minutes and think about what all this stuff means. Delta G, having a reaction be catalyzed by an enzyme, all that stuff. And then we'll come back and discuss it as a group. All right, so anybody think they have it? Yeah. So, okay, so let's start breaking that down. So what information do we have about this reaction? Yeah, so what does a negative delta G tell us? Spontaneous, yeah. So we know it's spontaneous, so we know the answer can't be D, right? What do we know about the speed of the reaction given just this information? So yeah, it's true that an enzyme will speed up the rate of the reaction compared to when there isn't an enzyme and it's uncatalyzed. But just from this information, we don't know whether it's fast or slow. We know that with an enzyme, it's faster than without an enzyme, but it still could be slow. We sort of just don't know enough from just this information. So that kind of gives away that the answer is C, right? So just from this information, all we know about this reaction is that it's spontaneous. And the fact that it's spontaneous, or that delta G is negative, just gives us information about sort of where the equilibrium lies, right? Once you set this reaction up, you're going to start to make a little bit of product, and then once you have a little bit more product than reactant, based on this small but negative delta G, then you kind of reached equilibrium. Yeah. To give us information about the speed, you'd have to be given. Yeah, so we'll talk more about that. I think it's in the next chapter about actual reaction kinetics and rates. But basically, you'd have to be given information about the reaction velocity or the reaction rate. And that can be in a number of different forms that we'll talk about, different ways to plot that information. But just from delta G, all you know is that the reaction is going to go in the forward direction, make some product, and you don't know how fast that's going to take or how long that's going to take. But like we said, it is faster with an enzyme than without an enzyme. Any other questions about that? All right, so let's start talking about different classes of enzymes and the different types of reactions they carry out. So we're going to do a few problems like this together, looking at example reactions and then asking you to figure out what type of enzyme, what class of enzyme will catalyze this reaction. So how about this one? Ribonuclease A is doing something to its reactant and making a product. Does anybody want to tell me what kind of enzyme it is? Yeah. Yeah, it's a hydrolase. How do you know that? Yeah, exactly. So water is one of the reactants, and we're using water to break this bond that's in the substrate, right? We break this bond kind of right over here somewhere. We end up with, we have one, and like a way you could write out this reaction sort of in general is you have one single substrate A, you add water, and then you end up with two separate products, right? You're cutting the substrate into two pieces using water. All right, so here is another reaction. So what is going on here and what kind of enzyme is doing this? B, yeah, it's a transferase. How do you know that? Yeah, exactly. Just like the name suggests, right? A transferase is moving one group from one substrate onto another substrate. So we have two substrates here. This methyl group is being taken off of one substrate and being stuck onto the other one when it's in the product. So in this case, our group is really small. It's just a methyl group. Sometimes the group can be larger, but the idea is you have two substrates and one piece is moving from one to the other one. So in general, you could write this reaction as A, B, where B is the group that's going to move, plus C, that's your other substrate. And then the result is you have A by itself, and then B attached to C in the product. All right, so here is our next one. What is happening in this reaction? What kind of enzyme is helping do this? Yeah. Sorry? Yeah, it's F. So we have an isomerization. So as you probably know, isomerization is when you have within a molecule a rearrangement of the atoms, the way they're bonded to each other. And so in this case, we have the substrate, but there's a rearrangement of something within the same substrate. So unlike a transferase, where a group is being moved from one substrate to another substrate, an isomerase is rearranging the atoms within the same molecule or the same substrate. So in this case, we have our glucose 6-phosphate with the phosphate group up here, and then our enzyme is helping to isomerize it to glucose 1-phosphate, where the phosphate is now on the 1 carbon. So a way you can write these kinds of reactions, is just A is turning into B. There, yeah, I don't know, not as descriptive as the other ones, maybe. Any questions about that one? All right. So now we have another reaction. So what's going on with this one? Yeah, it's a ligase. How do you know that? Yes, yeah, so you have 2 different substrates and you're joining them together and you're doing it along with ATP hydrolysis. So in this case, you have ATP is of pyrophosphate being removed, so it's turning into AMP plus pyrophosphate. You can also see it, so you can have ATP is being hydrolyzed here to AMP plus pyrophosphate. You can also see this with ATP where it's being hydrolyzed into ADP plus just phosphate, inorganic phosphate. Both of these are examples of ATP hydrolysis, right? You're cutting phosphates off of ATP, so you end up with adenosine with fewer phosphates on it. And that's along with the 2 substrates being joined together. So if you wanted to write out a sort of generalization of a ligation reaction, you have ATP plus A plus B, so you have 2 separate substrates, A and B, and then they're getting joined together into C with the 1 product, along with that ATP hydrolysis. And it doesn't always have to be ATP. It can also be another nucleotide like GTP, but pretty frequently we see ATP being used here because ATP is kind of the general energy currency in a cell that cells use for these types of reactions. Does anybody know the name of this particular enzyme? You kind of had a hint there where you said it was joining DNA together. It's okay if you don't. It's called DNA ligase. Very simple. So some of you may have encountered it like in the lab or somewhere, so just thought I'd throw that out there. Yeah, any questions about that? Yeah. I think in general it's nucleotide cleavage, and usually that's almost always ATP or GTP. I don't know about every single type of reaction. It's possible the other ones are also used, but in general, that's what you'll see most frequently. Yeah. Yep. All right. So let's go on to our next reaction. What's going on here? Yeah. So a carboxyl group isn't being removed. We just have we have our substrate that's being broken into two pieces. And something is happening while it's being broken into two pieces, but I will not give it away. Yeah. D, yeah, it's a lyase. Yeah, so what do lyases do? Anybody? So they break a covalent bond in the substrate, but they produce a double bond in the process. So it's a little bit like hydrolysis, except you're not adding in water to retain everything as a single bond, right? You're breaking the bond, and the result of that is you end up with a double bond because you're not like adding anything back into it. I don't know. For hydrolysis, it's kind of like if you think of like dehydration reactions from organic chemistry, like that kind of thing. But I don't want that to confuse anyone. But when you're looking at lyases, you should look for a substrate breaking into two pieces, but you're not, and the difference really, or one of the bigger differences, is that you're not adding in water as a reactant. Water is not helping you break the bond. So in this case, you have isocitrate. Make sure I draw this right. You're breaking this bond, right? So this sort of top piece becomes succinate, and then the bottom piece, you get a double bond here that gives you glyoxylate as your other product. So yeah, so like a generalized way you can write this reaction is A turns into B plus C. You're breaking A into two pieces, B plus C, but you are not adding water as a reactant. I do want to add that sometimes in these reactions you can like remove water, like that can be one of your products. So watch out for that. But water in that particular case, water isn't being used to break the bond, so it still is not hydrolysis. It's still a lyase reaction. And another way you can think of this as sort of removing a group. So you have isocitrate and you're removing a piece from it or a group from it to make two different products. So hopefully that helps conceptualize that in some way. Any questions about this kind of reaction? Yeah. Yes. Yeah. All right. So let's go on to our sixth one. So what is happening here? Yeah. Hey, yeah, this is an oxidoreductase. So it's an oxidation and reduction reaction. So in these types of reactions, you use an electron carrier that will either donate or accept electrons from the substrate. So if the electron carrier is being oxidized, the substrate is being reduced, or if the electron carrier is getting reduced, then the substrate is going to get oxidized. So in this case, we have NADH plus the proton is getting oxidized to NAD+, and at the same time we have our ketone that's getting reduced into an alcohol. So yeah, so so like here's one of our electron carriers, right? We have NADH and NAD+. Right. Can anybody think of any other common electron carriers we see in cells? Yeah. Just FAD, but yeah. FADH2 and FAD. Any other ones? Yeah. Yeah, exactly. When we have NADPH and NADP+. Got all those letters in there. All right. Any questions about that? Okay, so now that we've seen examples of all 6 of our classes of enzymes, I'm gonna put up these 3 reactions and give you all a few minutes. You have the same 6 options that we had from all the other slides. So take a few minutes, and for each of these 3 reactions, decide what class of enzyme is going to catalyze each of them. Yeah, all of these reactions will come back to haunt you. All right, so before we jump in, I do want to mention that these reactions really will all come back to haunt you. They might look random, but they're all parts of metabolism that we're going to be covering in some of the upcoming chapters. So you don't have to, remember them right now, but when you see them again later, might be why they look familiar later, because you saw them today. So let's start with the first one on the left. So fructose 1,6-bisphosphate is turning into glyceraldehyde 3-phosphate. What kind of enzyme is doing this? Yeah, lyase. Yeah. How do you know that? Yeah. Yes, exactly. Yeah, so we're cutting it right here. It's very helpfully color-coded it for you. So the red piece goes one way, the black piece goes the other way, and you have the formation of this double bond. All right. What about the middle one? Yep. So an isomerase is when you have a transfer of a group within the same molecule. So that's not quite what's happening here. Any other ideas? Yeah. Yeah, it's a transferase. I don't know where to write this. Transferase. So yeah, we have a transferase reaction here because we have one group that's being moved from one molecule to another molecule. So essentially what's happening here is this phosphate group in red is being transferred onto ADP, which is the other substrate. And then so ADP ends up as ATP, as the phosphate that it stole from the 1,3-BPG, right? And I also want to point out that in case anyone might have thought this was a ligase reaction because we saw ATP in there. It is not. It doesn't use a ligase because we're not attaching together two substrates using ATP hydrolysis. We're transferring a phosphate group from one substrate to another. Yeah. Yeah. I've never seen that happen. I'm not going to say it's impossible, but generally it's a lower, I think you get like essentially less energy out of breaking that bond from ADP to AMP compared to when you start with ATP. So I think that is why you don't see that as frequently. Yeah. Yeah. Any other questions? All right, so how about the reaction on the right with pyruvate becoming oxaloacetate? Yeah, it is a ligase. Yeah, so here we have a ligase because, like I said, like we said before, ATP is being hydrolyzed to help this reaction happen, and we're combining together our two substrates. So our CO2 is being attached on to pyruvate with a covalent bond to form oxaloacetate. And ATP hydrolysis is kind of providing some energy to help us do this. All right. Any questions about the 6 classes of enzymes before we move on to some other stuff? Okay, so so now we're gonna have this question just about reaction mechanisms in general. So there's a choice of 5 different amino acids here, and so I want you to think about, of these 5, which one or more than one is more likely to be a nucleophile to participate in covalent catalysis, and which ones might be, number 2, more likely to participate in acid-base catalysis. And before you start, I want to say this isn't like a very clear or definitive sort of rule. There's sort of fuzziness that we'll discuss as we go over the question. But sort of, this is just to get you kind of thinking about maybe the difference between these 2 types of catalysis mechanisms. So I'll give you all a few minutes to ponder this one. All right. Hopefully people are done, hard to tell. But so let's start thinking first about number 1. So which amino acid is more likely to be a nucleophile for covalent catalysis? So before we start picking out amino acids, what do we need for an enzyme or a group to act as a nucleophile? What characteristic does it need? Yeah, it needs what kind of charge? Negative charge. Yeah, needs a negative charge, or possibly just a lone pair of electrons. But it has to be able to carry out a nucleophilic attack, right? So for that, you need electrons. So now that we kind of have thought about what it means to be a nucleophile, which amino acids here have the possibility of doing that for us? Yeah. B, yeah, aspartic acid, exactly. So aspartic acid, in physiological conditions, at physiological pH, I should say, the side chain is deprotonated, right? So it's COO minus, so that's very handy. Yep. What about non-covalent catalysis? I'm not really sure what you're getting at. So we went over in the video, there's a few different types of other catalysis mechanisms we went over. So like there's like metal ion catalysis and like a whole variety of other things. But when you're talking about nucleophilic attack, you're generally talking about covalent catalysis. If that helps. Okay. We also talked about things like just helping to stabilize different transition states and things like that. Those are all different ways that enzymes can help catalyze reactions. There's just enzymes have a variety of things they can do to help a reaction proceed, and so the two we're going over right now are just covalent catalysis and acid-base catalysis. Yeah. So we have aspartic acid or aspartate, right? It has a negative charge, so it's able to act as a nucleophile, form covalent bonds potentially. Anything else on this list that might have a negative charge or a lone pair that might want to do covalent catalysis? Yeah, so histidine is an interesting one. So as you may or may not remember, the side chain of histidine has a pKa of 6, so it's really close to physiological pH. So histidine, you see it participating in, it can definitely act as a nucleophile when you have the nitrogen, right, has a lone pair. So that is certainly true. And when we go over acid-base catalysis, we'll also see it come up again, because, yeah, of other properties of histidine. But yeah, any other ones on here? Yeah. So lysine has a really high pKa. It tends to have more of a positive charge at physiological pH. It doesn't tend to be able to use its lone pair like for a nucleophilic attack. Yeah. Yes. They are, but I guess it's sort of complicated because you have to consider the sort of protonation state depending on what pH the environment is. So histidine, since it has a pKa around 6, is really easy to protonate and deprotonate. So there are times when it has a lone pair that's available, which could help it do nucleophilic attack, as compared to lysine, where it usually doesn't at physiological pH. It's usually protonated. Versus aspartic acid, which has a lower pKa, side chain is around 4. So it tends to be deprotonated around physiological pH, so it's more likely to have that negative charge. That being said, within the active site of an enzyme, the protonation states can be different than you might expect than just to see like if the amino acid was like floating around in the environment, which we saw in our example from this chapter, right? We saw a particular type of protease that uses its side chain to help do covalent catalysis, which is one of the amino acids in this list. Does anybody remember what that amino acid is? So yeah, in the second part of this chapter, we talked about a certain type of protease, right? And its catalysis mechanism. It's in the name of the protease. All right. If nobody wants to say it, I'll say it. So it's serine. So serine, the side chain of serine it's an alcohol group, right? And usually that also has a pretty high pKa. So like under normal physiological conditions, it's not going to be deprotonated. But within the active site of an enzyme, especially in combination with acid-base catalysis, that we'll talk about when we talk about serine proteases, the serine can get deprotonated, and so you have this oxygen with a negative charge, this oxyanion, and that is really unhappy by itself. It is very happy to do a nucleophilic attack and hope to form a covalent bond. So, yeah. So of this list, I would say probably aspartic acid and serine are probably your best ones for nucleophiles and covalent catalysis. And histidine is kind of like a mix, but yeah, we'll talk about that in a moment. So first, let's talk about acid-base catalysis. So we talked about for covalent catalysis, we have a nucleophilic attack. What's happening in acid-base catalysis? Think of your Bronsted-Lowry acids and bases. What are they doing? What's that? Yeah, exactly. They're donating or accepting a proton. And so, like I was sort of hinting at, which one of these amino acids is pretty good at doing that? So lysine, again, has that really high pKa, so it can, but it's harder, right, for it to do. But it's possible. I won't say it's impossible. Which other ones? Which one of these amino acids has a pKa that's really close to physiological pH? Yeah. Sorry? Yeah, exactly, histidine. So histidine is really happy to donate and accept protons. It loves doing acid-base catalysis. We also see an example of that in our serine protease when we talk about that mechanism. Yeah, that's histidine. Is there anything else on this list that might be willing to donate or accept a proton that you think? So serine, like I said, it really doesn't want to just donate away that proton. It really needs like help to do that because it has that high pKa. Anything else here that has? Yeah, exactly, aspartic acid. So aspartic acid can possibly get protonated or deprotonated. It has a pKa around 4. It's a little bit happier to do that than serine or lysine. But I would say for acid-base catalysis, your top choice is almost always histidine from this list. So that brings us to our last amino acid, phenylalanine. It doesn't want to do either of these things, right? It's just an aromatic ring. It really doesn't want to donate or accept protons. It really doesn't want to participate in nucleophilic attack. It's just sitting there being aromatic. I don't know. Any questions about these two types of catalysis and how you might differentiate between them? Okay. So that brings us to the, yeah, yep, yeah, pretty much. Maybe, yeah, kind of. It's less likely, but I won't say it's impossible. Yeah. Yep. Yeah, so Sirian, you have to really convince it to lose a proton. But once it loses a proton, it's a really good nucleophile. But it won't just like donate away the proton. Yeah. Yeah. I would say B is probably better at 1 or maybe both. I don't know. So like, I mean, if you're thinking about pKa's, right? pK for histidine. Oops, turned off my pen. What is happening? Sorry. So the pKa is about 6. pKa is about 4. So like kind of the closer you are to the pKa, the easier it is for it to kind of fluctuate between donating and accepting protons. That's one way to think about it. And physiological pH, as you know, is a little above 7. But like I said, there isn't really, you know, a strict rule. Like it can only do one or it can only do the other. You can kind of argue either way for a lot of them. Okay, so let's move on to serine proteases, which hopefully we can get through this. So yeah. So let's talk about serine proteases. So just off the top of your head, can anybody tell me what serine proteases are doing, what kind of catalysis they're doing, what kind of bonds they're working with? Or do you need a few minutes to look through everything? Okay, I think everyone needs a few minutes. So I'll give you all a few minutes to look through your notes from serine proteases, and we'll come back as a group. right, so let's at least start with the first part of this slide. So what do serine proteases catalyze? What kind of reaction are they doing? Yeah. Yeah. So they catalyze the cleavage of peptide bonds in what? What is the substrate? Proteins, yeah. And you could also say it a little bit differently. So if you wanted to be a little bit more specific than cleavage, you could say hydrolysis. Both are correct. What is a more general word for peptide bonds? What type of bond is a peptide bond? Amide bond. And what is another word we might use rather than protein to describe a polypeptide? Yeah. Cool. Yeah, so these are, for each of these lengths, either of these answers is completely correct. So let's try the middle part. So what determines the location of cleavage within the substrate? Yeah, the specificity pocket. And what is the name we use for the reactive amino acids in the active site? Yeah. Yeah, the catalytic triad. And what part of this enzyme helps to stabilize the tetrahedral intermediate? Yeah. Yes, the oxyanion hole. Cool. So the last part of this slide is about the 3 different amino acids that are in the catalytic triad. So I guess first of all, can anybody tell me what 3 amino acids are in the catalytic triad? Yeah. Yeah. So the catalytic triad is histidine, serine, aspartic acid. So of these 3 amino acids, which one of them is going to carry out nucleophilic attack on the substrate? Yeah, serine. So just like we talked about in the last slide, serine is going to get deprotonated, and then it is very upset, and so it carries out a nucleophilic attack on the substrate. So, yeah, so when it, I guess that kind of brings us almost to the last one, right? So if serine is carrying out nucleophilic attack on the substrate, which enzyme is responsible for covalent catalysis? Yeah, it's also serine, exactly. So, yeah, okay. And so when after serine carries out the nucleophilic attack, what forms? What kind of intermediate forms? Yeah. Yeah, and so what is tetrahedral referring to? Yeah, it's a carbon with bonded to 4 different things, right? And so So that's what we mean by covalent catalysis, right? We went from a carbon that was bonded to 3 different things to a carbon that's now bonded to 4 different things. We made a covalent bond. We did covalent catalysis. Okay, so let's go back to the second one. So which one of these amino acids helps to maintain the geometry in the active site? Got a 1 out of 3 chance. What's that? Histidine? It's not histidine. You got a 1 out of 2 chance for the other ones. Yeah. Aspartate, yeah. So, yeah, aspartate has a really important job, which is to maintain the correct geometry of the active site. It's not participating directly in the acid-base and covalent catalysis that we see, but it's still there. It's part of the catalytic triad, and it has its own job, which is mostly helping to maintain geometry. All right, so what about our next one? So which one is donating and accepting protons to and from serine? Yeah, that one's the histidine, yeah. And so what kind of catalysis is that? Yeah, so this is the acid-base catalysis we were talking about. We're donating and accepting protons. We're carrying out acid-base catalysis. So which amino acid is also donating and accepting protons from the substrate? It is not serine. It is not aspartic acid. It is also histidine. Yeah. Got a 1 out of 3. Yeah, so histidine in, where was I going with this? In the active site, in this catalytic triad, histidine is really doing all this acid-base catalysis. It's donating and accepting protons, and it does this a couple times. Serine is really only doing covalent catalysis. So let's look at that in more detail, since maybe we need a refresher. So yeah, so when this ends, so just to orient everyone, so our substrate is shown here in blue, the polypeptide, and it's just showing us the peptide bond, right? The rest of the protein is just R groups. And so ultimately the goal of this enzyme is to break this peptide bond using hydrolysis. That's what's going to happen. And it goes through a whole bunch of steps to do this, right? So it doesn't just immediately break the bond. So before it can get there, first we have general base catalysis using histidine, right? So the nitrogen on this histidine has a lone pair. It accepts an electron from serine, and this lets the oxygen from serine carry out a nucleophilic attack on the carboxyl end of the protein. That's the substrate. So a couple of things happened. We had base catalysis and nucleophilic attack, and then this helps us to form this nice little tetrahedral intermediate. And again, it's tetrahedral because now this carbon has bonds to 4 different things. And it also has this oxyanion, which makes it pretty unstable. So this is one of the points where the oxyanion hole that's in the enzyme is going to help to stabilize this intermediate. But eventually, the tetrahedral intermediate is going to collapse back down. And as it collapses back down, we have the movement of electrons, ultimately breaking finally this peptide bond. And then histidine is again now histidine is going to act as an acid, right, and donate this proton. So we end up here at this acyl enzyme intermediate. So now we have, what we sort of wanted to happen or expected to happen was that our peptide bond broke. So at this point, our new N-terminal end of our peptide is only attached to the active site of the enzyme by a hydrogen bond. So it is able to diffuse away. It's not covalently bound anymore. But we still have a problem because the new carboxy terminal end is covalently attached to the enzyme. So not done yet, right? We had to finish because we had to release the other end of the product. And so essentially we have almost a repeat of the same thing again. So now we have water is finally floating in, right? This is a hydrolysis reaction. Water has to get involved. So once the N-terminal end of the protein floats away, now we have water coming in to kind of replace it. And then histidine does the same thing again. So now it's going to act once again as a base. It's going to take that proton from the water. Water is now going to carry out nucleophilic attack on that same 4 carbon. Now that 4 carbon is once again in this tetrahedral intermediate, and it is again unhappy. And so again, the tetrahedral intermediate is going to collapse. Once again, histidine is going to act as an acid and donate away its proton. And this time we can finally break the bond to the serine that's in the active site, so that finally the new carboxy terminal end of the protein can float away, and the catalytic triad is reset to its original state. So overall, we had histidine did base acid catalysis, serine acted as a nucleophile, water also acted as a nucleophile, and we broke a peptide bond. Does anyone have any questions about that mechanism? I know it's a lot. It is also in the video if you haven't watched it yet. All right, I'll let that simmer. If you have any questions, you can always ask me later. So let's move on to some of our examples of serine proteases. So also in this chapter, we talked about 3 different examples of serine proteases that are digestive enzymes, right? We have elastase, trypsin, and chymotrypsin. So in this question, we want to figure out which serine proteases would cut each of these bonds that are identified with arrows. So there's 2 different bonds that might be cut. Different serine proteases are going to have specificity to each of these cut sites because of their selectivity pocket, right? So, so which amino acid helps decide? So like if you're looking at this first arrow that's drawn here, which amino acid is going to fit in that specificity pocket that helps that protease decide to cut there? Before the cut or after the cut? Yeah. So, okay, so before you decide which enzyme it is, are we looking at the amino acid before the cut or the one after the cut to make the decision about which enzyme it is? Before, yeah. So look before the cut site. So for this first arrow, which amino acid is before the cut site? Might be a silly question, but gotta ask. Alanine, yeah. And so of these 3 protease enzymes, which one is going to be more likely to cleave after alanine? So it is not trypsin. So what characteristics does alanine have? Is it large? Is it small? Is it charged? Is it uncharged? It is small, but it's uncharged. So the alanine side chain is a methyl group, right? Yeah. So this is the N-terminal end of the amino acid. Here's the carboxyl terminal end. So first of all, we know alanine is before the site because we're going from N to C from left to right, right? So always double check that. And so this methyl group is small, it's uncharged. So we know it's not trypsin, but which one of these other 2 might cut after a small and uncharged amino acid? It is not chymotrypsin. It is elastase. Excellent work. Yeah. 1 out of 3 chance. You're going to get there eventually. Yeah. So elastase cuts small after a small uncharged amino acid. So what about our next arrow? So what amino acid is right before that cut site? Phenylalanine. Yeah, so what characteristic does phenylalanine have? Yeah, it is bulky. Whoops. It is, I can't spell anymore. It's bulky. It's hydrophobic. It's aromatic. So which one of these serine proteases is it going to be? It's not elastase, which we know cuts after small uncharged amino acids. So of the other two, anybody want to throw a guess out? Yeah, it's chymotrypsin. Yeah. Okay, so that brings us to our last one, which is trypsin, that we don't have an example for, obviously, but does anybody have an idea of what trypsin is looking for right before it's cut site? Yeah, charged. Positive or negative charge? It's positive charge, yeah. Yeah, 50% chance of positive or negative. Yeah, so positive charge. All right, so those are our 3 digestive proteases that we talked about. They are each selective to cut after a different type of amino acid. Okay. Any questions about that? Cool. All right, so now you just get to listen to me talk about SARS-CoV-2. You're off the hook. So we've seen, so yeah, once again, it's our favorite virus, SARS-CoV-2. Always got to start with that. And we've seen this diagram before. I think it was in chapter 9 when we were talking about how it's really important to cleave the spike protein into 2 pieces so it can enter into our cells, right? And now we were just talking about enzyme mechanisms. We're cleaving a protein, right? We have protease is happening, right? And so we have, in this picture, we have furin, right? Furin is a serine protease. Fun fact. So, yeah, so now that you know all about how serine proteases work, here's a real life example of one. Although this one actually works a little different than, or maybe, yeah. Sorry, I shouldn't say it works differently. The specificity is different. So we talked about our digestive proteases that have a specificity pocket that looks for one specific amino acid right before the cut. Furin is a little bit different. It has a recognition sequence that's a few amino acids in length. So a typical furin cleavage site, it's called a polybasic site because it has a bunch of basic amino acids in it. It has an arginine followed by anything, then followed by a couple more basic amino acids, lysine or arginine, and then another arginine. It's interesting because SARS-CoV-2 is able to be cut by furin, but the recognition site is a little bit different. So it's 2 basic amino acids followed by an alanine, and then just one more basic one. So the very similar, but like a little bit different than sort of a typical furin cleavage site. And so there's been a lot of talk about this, right? So something that you may find interesting is that the original SARS coronavirus that was around however long that was, almost 20 years ago now, does not have a furin cleavage site in it. So this is new for SARS-CoV-2. And so this has given some people the idea that maybe this furin cleavage site was like engineered into the virus to make it more like virulent or make you sicker. And that's kind of one reason why people have this hypothesis that like this virus was, designed in a lab and like released to cause a pandemic. But But I ask you to look at this furin cleavage site, right? It's not a typical furin cleavage site. So one question you might ask in response is, why would you design in a furin cleavage site that's like not ideal for actually being cleaved by a furin enzyme? Seems like a weird choice, in my opinion. So if anyone, I don't know, has any thoughts about, is this, there's, one camp of people that, thinks maybe this virus was designed in a lab, another camp of people that, thinks there's better evidence that the virus, arose in nature through selection. I think to me this is a pretty good sign that no one would do this on purpose. Seems kind of weird to do this on purpose. So that's kind of something interesting you can think about in terms of SARS-CoV-2 and cleavage by a protease. I also want to add that in other viruses, furin cleavage sites have been shown to make them a little bit more pathogenic, like pandemic flu viruses often have furin cleavage. But we don't know for SARS-CoV-2 in particular if being cleaved by furin makes it more pathogenic. It's just there, and that's how it works, but we don't know if that makes the virus better or worse at doing its job. So you had a question.
So, furin.
Yeah.
Okay, it's not.
No. Yes. Yeah. So furin is in our cells or on the surface of our cells, and it is. So just as a reminder, we have our pre-fusion spike protein. We have our ACE2 receptor. Once the spike protein interacts with the ACE2 receptor, that signals for furin to cut between the S1 and S2. The S1 stays attached to the ACE2 receptor, and the S2 goes through that conformational change that helps into that helical little shape that helps it to penetrate the cell membrane and help it enter. And so the cleavage is required for the virus to enter our cells. It's pretty interesting. So, and now we know how it does it, because we all know how serine proteases work. Any questions about that? There's also some links here if you're interested in furins and how they work and what they're doing in the context of infection. And so that's really all I had to say about enzymes and serine proteases. So just another reminder, like I said at the beginning of class, if you haven't submitted your project update yet, please submit before tonight. We're going to do Chapter 12 on Thursday, so please be prepared to talk about Chapter 12 on Thursday. And we just finished Chapter 11 today, so the quiz 9, which is about Chapter 11, is due a week from today, next Monday night. And I think that's all I have to say for you. Yeah. Yes, that will be the Monday after Thanksgiving, because I didn't want to make that due on Thanksgiving. Yeah, that would be rude. Yeah. I'm not that cruel. Yeah. So if anybody has questions for me, I have office hours. You know how to find me, and we'll end there for today, because I don't think anyone wants to talk about Chapter 12 today. So.